In this podcast, we are going to give a very basic introduction to how DNA replicates itself. All right, so DNA replication is easily confused with protein synthesis. So let's talk a little bit first about when DNA replication is going to occur. Anytime any of your body cells or your somatic cells need to copy themselves or reproduce, or say you have an injury and you need to produce new cells, if you're making an exact copy of a cell, you're going to need to copy your DNA for that brand new cell. That's when replication occurs. Think of it as being almost like a Xerox machine copying an entire set of directions or a cookbook. Replication requires both strands of your DNA to be separated, and then each side of your strands is going to serve as a template strand or a pattern for duplication. In this sense, we consider DNA replication to be semi-conservative. So looking at this diagram, we have two blue strands. Each of these come from our parent DNA strand. So there are two sides, and you can see they've unwound it here in this example. You have your parent strand. Each one is complementary to each other. A pairs with T, G with C, etc. So in replication, we're going to take those two strands and we're going to separate them. And when we do that, here's our original strand from the right. Okay, we're going to start with the parent strand, and then we are going to add nucleotides that are complementary. So we're essentially going to rebuild the side of the DNA that is missing. So in this case, we have one new strand, which is shown in red, and then this blue is our template strand number 2. And on the other side, we have template strand number 1, which came from our parents, and we're going to add complementary bases. So we're going to have a new strand as well as a parent strand, or a template strand and a new strand, however you want to refer to those. So again, if you were to look at these as a whole, these strands of DNA, along with the bases and the order of bases, it's going to perfectly match. It will be an exact replica of our original strand. But one of the strands, and that's shown in blue, is conserved from the parent DNA, and one strand shown in red is going to be new, or the daughter DNA. All right, so DNA replication, it should be noted, is much more complex, but we're going to give just a general overview for you to get the right idea of how DNA replication occurs without going into all of the nitty-gritty details that you wouldn't need per se in nursing. All right, so replication is going to begin when you have a helicase, and remember that ASC tells us it's an enzyme, and the function of helicase is to unwind that double helix. And it's going to do this by breaking hydrogen bonds. It's also going to start to separate your two strands of DNA. So imagine on this diagram that your helicase is right here, and it's going to move up the strand, and it creates what we call a replication fork. And the reason it's called a replication fork, and I'm going to have to scooch back for this, is because your two strands are together, and then the enzyme comes up and it separates those strands. Or it's, if you will, unzips those genes from each other. So we need helicase to unwind and to separate the two sides of our DNA from one another. The second step is to bring in an enzyme that can add DNA nucleotides to each of the parent strands. This enzyme is DNA polymerase, and looking at this name, we know it's an enzyme because it has that ASE ending, but then look at this. This is an enzyme that builds polymers of DNA. So DNA polymerase adds complementary nucleotides, complementary bases to the parent strand. So it essentially builds a whole new side of that daughter strand so that we have two full strands of DNA that are exact copies of each other. DNA polymerase also is going to catalyze the formation of phosphodiester bonds between nucleotides. So if you remember from previous lectures, and I'm going to do just terrible drawings here so you know what I'm talking about, except for we're going to label these with P's, PSP. The backbone of our DNA are going to be alternating sugar phosphate groups. And so DNA polymerase is going to come in and it's going to catalyze the formation of that phosphodiester bond between nucleotides. So it's going to help to build our new strand of DNA. So on earlier slides, we talked about this idea of helicase coming in and creating a replication bubble and replication forks. So imagine that this strand that you're looking at was a DNA that was unwound, it's laid flat, and now we have a helicase that can come in and pull those two strands apart. It separates the hydrogen bonds between nucleotides and it creates two replication forks. Those little green circles are where the replication forks are. Eventually, as you are creating the complementary strands, those replication bubbles are going to get bigger and bigger and eventually run into each other. And once they do that, you're going to wind up with two separate identical strands of DNA. So wherever that helicase comes in and separates the DNA, we call that the origin of replication. That's where it starts. Because this is not just simple and easy, we wind up with an issue. DNA can only be replicated in the 3' to 5' direction, meaning, as you know, these are anti-parallel, they run in opposite directions. So we can only replicate continuously from the origin of replication in the 3' to 5' direction. This creates a leading strand. So notice from the origin of replication, I have a continuous strand on the top right here. Well, since we're running anti-parallel, here's my origin of replication. I will also have a leading strand going in the opposite direction from the origin of replication. The issue is behind, in the space behind the origin of replication, I can't go backwards. I can't start at the origin of replication and go in the 5' to 3' direction because that's not the direction of replication. And what gets created is called a lagging strand. So we have these primers that come in, and the primers are really just a spot that says, hey, DNA polymerase, come and add sections here. So we add these little primers. DNA polymerase comes in and it is able to synthesize these little tiny sections or these fragments of complementary DNA. So we wind up with these little sections and it will build these until you get to the primer. Eventually we take that primer out and we seal all of it together with DNA ligase. We call these the lagging strand and we call these fragments specifically the Okazaki fragments. So big takeaway for this is that DNA replication is more complicated than we have time for, but you will have a leading strand and it always builds in the 3' to 5' direction, and because of that you have also a lagging strand that is put together in smaller fragments.